Telomeres and stem cell aging in regards to CRISPR-Cas9. Telomere are the protectors of the universe. They lay within each cell of the eukaryotic species. They are located at the terminal end field of each arm within the X-shaped structure of the chromosome structure where the majestic DNA strand lie. Telomeres consist of these sequences of each end terminal and provide unique mechanisms for the faithful replication of linear DNA molecules. DNA polymers cannot synthesize DNA to the very ends of a linear chromosome. We call this the end replication problem. Each single characteristic and variation of telomere length are affected by genetic and non-genetic factors. Here is a schematic overview of telomere lengths as it pertains to cardiovascular disease. Critically short telomeres lead to cellular apoptosis or dysfunction. This contributes to atherogenesis and weaken and reduce repair and regenerative capacity of the stem cell production line of the cardiovascular system. Disease-promoting factors such as smoking accelerate telomere shortening through inflammation or increased oxidant stress. However, disease-protective factors such as exercise can activate telomeres actively and maintain telomere strength and length. How do we solve the end replication problem and how are they replicated, you ask? The end replication problem causes telomeres to shorten over successive cell divisions. Solving the end replication problem is a key function of telomeres, which are replicated by the enzyme telomere. First, let's learn about key aspects of cell division and central dogma. If you look closely, you will see that the chromosome is wearing a fancy hat. The hat is called a telomere and it allows them to copy DNA safely. Each time I divide, my telomere gets a little shorter. When telomere becomes too short, it cannot divide anymore and it causes apoptosis. Artificial chromosomes provide a means of better understanding the functional significance of this feature. Researchers have constructed human artificial chromosomes, HACs, and may eventually provide new avenues for the treatment of genetic disease with stem cell therapy. These ongoing efforts to improve our scientific processes of correcting disease in the human somatic cell line have promising results with the majesties of the CRISPR-Cas9 system. This genome editing tool that one might imagine as a pair of scissors is programmed in nature and occurring as a phenomenon in cells that the books belonging to the most prestigious universities cannot even explain. One might imagine it as the highest form of intelligence with derivatives originating from other dimensions. At least I would. These imaginary scissors that one might imagine are guided as an energy force vector. Clustered regulatory interspace short palindromic repeats, CRISPR, is essential within a single strand of guide RNA. gRNA is a non-coding short RNA strand that acts as the force vector which first guides CRISPR. These enzymes are composed of genes organized in operons and serve as genome targeting sequences called spacers. This system of mechanisms together are CRISPR-Cas. They bind to the host chromosome at the proximal end of the CRISPR array. In the expression and interphase phases, transcription of the repeat spacer occurs and thus precursor CRISPR RNA or pre-CR RNA molecule is formed. Enzymic cleavage yields the short CR RNA that pairs with complementary protospacer sequences of plasmid targets. CrRNA targets recognition of the precise genomic sequence to which the splicing is needed. Now that we have found the precise mutation and location, splicing of the point mutation is carried out by protein. Cas9 plays the vital role of the splicing action and is heavily utilized in the genetic engineering applications. Its main function is to cut the DNA, hence altering the cell's genome. Here, Cas9 splicing is guided by CrRNA, tracer RNA. forms a RNA structure that directs the CRISPR-Cas associated protein Cas9 to introduce double strands that breaks in target DNA. In the original research paper, Jennifer Doudna and Emmanuel Carpentier found that Cas9 endonuclease can also be programmed with a single chimeric RNA. The possibility of a single RNA guide Cas9 is appealing due to its potential utility for programmed DNA cleavage and genome editing. This single transcript effectively fuses the 3' end of the CRRNA to the 5' end of the tracer RNA, thereby mimicking the dual RNA structure required to guide site-specific DNA cleavage by Cas9. They found that rational design of chimeric RNA is robust and could in principle enable targeting of any DNA sequence of interest with few constraints. This milestone has formed a tool and marker in history for all women, and that is the first time in history that not only one, but 2 women have won the Nobel Prize. It is currently the most advanced form of biological, I'm almost done, engineering with cutting edge technologies. It is just in its infancy, revolutionizing our world of medicine as we know it today. No, that's okay. It was really long. Okay, I'm done. Are you sure that's it? Okay.
And I'll text you that information for the sale.
Do you guys want cash? I have cash, or do you prefer Zelle?
It don't matter. You have a-
I have cash.
Okay.
Oh, you didn't take a picture actually. I do. I have your number now. Okay, look at everything. Does it look straight to you? Did you guys use
No.
oh, you did? Oh, okay. Oh, okay, let me see this.
You live here alone? I live on the other side of the freeway, so I come over here.
Oh, really? They're not bad. We pay like $1,300. It's a 2-bedroom with an office.
Oh, that's cool.
Yeah, we use the office as a room though. Thank you so much.
No problem.
And I will be calling you in the future. All right, you guys have a good day. Thank you.
This site seeing Stone Cold Steve Austin and Rich to the America to push the cold water washes with time save you energy. And it saves you up to $150 a year on your energy bill. Plus, Tycoon better and cold than the bargain brand hot. So what are you waiting for? Where to go? Because Stone Cold says so. Turn to cold.